All right, so part two of our video lecture, I promise this one is going to go a little bit quicker. There's a lot of foundation pieces that we need to get in that first part of really understanding cells. So this next section, we'll go a little bit faster. So we ended with our first slide talking about organelles, and we actually have two sets of organelles that we need to go to. So we ended up with the vacuoles. Go ahead and find the second page for organelles, and that one starts with vesicles. Vesicles are going to be another circular structure that you have within the cell that is going to hold and contain different types of liquids within the cell. And so it does similar to what the vacuoles do, but a little bit wider variety of materials that it can hold within those vesicles. And again, just working to contain those excess liquids so they don't interfere with any of the chemical reactions that need to take place. The next one that you have are the microfilaments. And microfilaments are going to, they're basically like little toothpicks. That's what they look like. They're made out of protein and they're a rod-like structure, toothpick-like. And what they do is they work to kind of mimic our skeleton, but they're very different as well. So our skeleton is there to protect. It also gives us shape and form. And the microtubules or microfilaments are going to do that as well. They usually are going to form a ring around the outside of the cell. So you have the cell membrane made out of those lipids. And then just inside that, you would have these protein rods that would line up and form a structure just on the inside of those lipids. Now, the unique thing about this is they line up just inside that lipid layer to offer protection and help with the structure. But unlike our skeleton, they actually are able to disassemble and reform somewhere else in the cell. So if it is needed for maybe the cell to move, they can break down and reform in another part of the cell if it needs a little extra protection, or if you want to kind of make that part of the cell a little heavier to get the cell to roll or turn. And then they can reform back to their original shape once that job is done. So our bones are not able to come apart and reform somewhere else if we need added protection somewhere. They're in their set point. So microfilaments, which work somewhat like our bones, but also have the ability to break down and reform or rebuild somewhere else in the cell when needed. The other structures that we see on the cell are flagella and cilia. And in some ways they're similar, and in other ways they are very different from each other. They both are going to be these tail-like extensions that you have off the surface of the cell. So they're found on the outside of the cell. And with the flagella, you typically, if you have a flagella, it's one and it's very long. And the reason that you would have that flagella is going to be because the cell uses it to move. So the tail has a whip-like action. It flips back and forth and actually propels or moves the cell forward. So flagella, one, usually very long and used for movement. Cilia, on the other hand, are usually many. So if you have them all over the surface of the cell, and they are short, and they usually are going to be used as a cleaning mechanism. So you find them in places like the respiratory tract where you're inhaling dirt and other particles, and they just kind of flip back and forth to make sure that that dirt and those other debris particles can't build up around the cells in your respiratory tract. So they just flip back and forth, keep things moving to keep those areas clean. So both on the outside, both are going to have a wave-like action where they flip back and forth, but flagella were more moving and cilia were doing more of a cleaning job. From there, we have the Golgi apparatus, and the Golgi apparatus is going to be another area where we have these membrane-like sacs, which was the same type of description we had with the endoplasmic reticulum. So when you look at these on a picture, they actually can look very similar to each other. With the Golgi apparatus, the one thing that you can use to distinguish it and tell it apart from the endoplasmic reticulum is at no point does it ever touch the nucleus. So if you have these membrane-like structures and you're trying to consider whether or not it's the endoplasmic reticulum or the Golgi, just track it back to its origin. If you trace it back and you see that at some point it touches the nucleus, it is the endoplasmic reticulum. If you look at it and you can see those membranes, but it's contained and it's moved away and at no point does it ever touch the nucleus, then you know it's the Golgi apparatus. Now, the Golgi apparatus has a very special job. What it does is it actually takes those products that were made within the cell and it packages them for their final destination. So sometimes it's considered to be like the gift wrapper of the cell, because when these products are made and then they have to be transported somewhere else in the body to be used, it's kind of like if you're getting ready for a trip. You don't just jump in the car and go. You're going to get some things ready. So you're going to have spare clothes, you're going to have some money, you're going to fill up your car with gas, you're going to do all of those things to prepare for the trip. And when these cell products are getting shipped out, they do the same thing. They have to get prepared first. And that's the job of the Golgi apparatus, is to determine exactly what they need and then provide them with those materials to prepare them for the trip. So if they are going somewhere where they need protection, Golgi apparatus is going to attach some lipids, some fat for that protection. If they're going somewhere and they're going to be using a flagella to move them there, then the Golgi apparatus is going to provide them with some carbohydrates so that they can use that as an energy source and keep that flagella tail moving back and forth. So if they're going to be used in a place in the body where they're actually going to be building more material, Golgi apparatus will package it with protein so that it has that protein to then build the new tissue. So depending on what that cell product is going to be doing, Golgi apparatus will figure it out and then attach the right supplies to that cell product before it gets shipped out. The next one that we have on the list, if anybody has taken a biology class, I have found that this one tends to be the one that sticks in people's brains. I say the mitochondria, and immediately people will kick out powerhouse of the cell. And like I said, for some reason, that connection just seems to be a very strong connection through educational backgrounds. The mitochondria is a very special organelle because it is going to provide the energy that a cell needs in order to be able to do work. So we talked about ATP production and the process of cellular respiration, and that you have glycolysis, you have Krebs, you have the ETC, and when we look at the ETC producing those 32 ATP molecules, the one thing that I didn't mention, and the reason I didn't is because I knew we were going to pick it up later on, is that the ETC actually happens inside the mitochondria. So within that mitochondria, we talk about it as being the powerhouse of the cell and supplying energy for the cell, because that is where that mass production of those 32 ATP take place, is within the mitochondria. Again, the cells will differ in the number of mitochondria that they have. If you have a cell that is going to need a lot of energy, it has a high number of mitochondria. If you have cells that don't have high energy demands, they don't have as many mitochondria in them. But your cells, most of your cells, do contain mitochondria. And again, there's always an exception to the rule. We'll get to that later. Centrosomes, I'm not going to spend a lot of time on these guys because we get into them when we talk about mitosis, or the process of cell division. And so centrosomes play a very important or key role in the process of cell division, and we'll come back to that later. The next one that you have, or the last one on the list, are lysosomes. Lysosomes are another circular structure, and they actually have something a little bit stronger than just simply water or random liquids in them. They are going to contain digestive enzymes. So a lot of times when I introduce lysosomes, I refer to them as being the garbage cans of the cell. So within the lysosome, they contain this digestive enzyme. And when you have things that are damaged in the cell, or when you have things that are dying in the cell, they're going to get delivered to this lysosome. So if you have something that's broken at home, you usually throw it in the garbage can. Same thing happens within the cell. It gets delivered to the lysosome, and the lysosome will ingest it, coat it in that digestive enzyme. And basically what the digestive enzyme does is it eats away at it until it's broken down, degraded. And essentially what it's doing is it's making sure that you don't have all of this waste that is building up inside the cell. If you think about it, you guys have garbage collection that comes once a week. Imagine if it didn't happen on a weekly basis, but it happened on an annual basis, and what your homes would look like if you had to store all of your garbage somewhere in your house until that once a year pickup came. Your houses would be very cluttered, very dirty, a lot of problems. It would be unlivable in some cases after a while. And so the lysosomes are going to make sure that that garbage gets collected, it gets broken down, destroyed, removed, so that it doesn't build up in the cell and potentially cause cell death. So the next ones that we have talk about how we move things across the cell membrane. And there are really two overlying ways that we move things through the cell membrane. One is by passive transport and one is by active transport. Now, passive transport means that we're able to move it across the cell and we don't have to use energy to do it. Active transport, we're moving things across the cell membrane, but we have to use energy in order to get it to happen. Now, the thing that's important here is, again, apply this to what we were talking about earlier. If you had to supply energy every time you wanted to move things across the cell membrane, and consider that one cell is going to move hundreds of things in and out on a daily basis, and then you multiply that by the fact that we have trillions of cells in the body. If it took energy every time you wanted to move something, and you map that out or multiply that out over those trillions of cells, there is no way you would be able to eat enough to get enough calories into your body to keep up with that energy demand. And we would not survive. So we would either have to be made of a lot fewer cells or we wouldn't live. So a majority of the cell movement that we use in the human body is going to be by passive movement. It does not require energy in order to happen. That is key and that is very important. Now, passive movement is, like I said, no energy, but the molecules are going to move from an area where they are highly concentrated to an area where they are in low concentration. You guys see this all the time. If you've ever had a cup sitting on the kitchen table and you knock it over, while that cup is upright, you have, let's say you have water in it, you have a high concentration of water molecules inside that cup. When you knock it over, they don't stay in the cup because that's where they're highly concentrated. What do? They spill out to an area where they're in low concentration, and they will continue to spread out across the table until they reach equilibrium, which means until their molecules in the water are separated out from each other. So when we talk about this, anytime we're referring to passive movement, high to low concentration, that is the way that the molecules are moving. Again, if you've ever used air freshener highly concentrated in the container. When you spray it, you release them from where they're highly concentrated out into the room where they're low concentration. And so that's why they will spread out and drift throughout the room. And if you spray it in the back, after a couple minutes, anybody standing in the front of the room will eventually be able to pick up that scent because they are moving to an area where they are in low concentration. So the types of passive movement that we have, first one is diffusion. No energy, high to low concentration. And when we talk about diffusion, we're talking about moving molecules that are anything but water. So water gets its own name, but any molecule other than water that is moving through the cell membrane is going to be, we're going to refer to that as diffusion. Facilitated diffusion is a little bit different. Facilitated diffusion, you facilitate something, you have somebody helping or aiding the process. So facilitated diffusion is for those molecules that are a little bit bigger and they can't just simply move between the lipids. They have to use those protein channels because they are larger in size. So diffusion, they're moving in between the lipids. They're the small little molecules. They can pass in between, no problems. Facilitated diffusion, that's anything that's going to need to use those protein channels to get from point A to point B. The other one that you have is osmosis, still not using any energy, but the only time that you can use the term osmosis is when you are talking about the movement of water molecules. Cannot apply at any other time. So if water molecules are moving across the cell membrane, then you would refer to that as osmosis. Last one is filtration. Filtration, we'll pick this up again when we get into our excretory chapter. Doesn't require energy, but this time we are moving a particular molecule through a pore. So there is a designated opening within a structure for that material to move in and out. So it's a little bit different than what we have just simply passing between the lipids of the cell membrane. You kind of think of a sponge and all the little pockets and openings that you have in a sponge. That's more along the lines of filtration. In order to assist with filtration, we also use pressure. So if you, again, have ever used a sponge, you dunk it in some water. What do you do when you bring it up out of the water? You squeeze it. You apply that pressure to get the water molecules then to move out of the sponge. The next one that we have is active transport. And active transport is going to be the complete opposite of passive transport. So this time we are using energy in order to move the molecules from point A to point B. And this is because you are moving against the gradient. This time you are taking molecules from where they're in a low concentration and you are moving them to an area where they are already highly packed or highly concentrated. Molecules don't want to do that. They want their personal space. They want to be spread out from each other. So that's why it takes energy. You have to force them from where they're in small numbers and force them to be where they're already in larger numbers. Now, where would this be beneficial in the human body? Two main places are going to be your muscles and your nerves. Both of those are places where you are using ions, so molecules or atoms that have charges on them, and you're trying to take a bunch of positively charged molecules or atoms and pack them in together. Well, you know that positively charged atoms or molecules are going to repel, they're going to push against each other. And that's where the energy is going to come in, because you are going to have to take them and force them into a space together, even though they are trying to push and repel off of each other. So within your muscles, you have to pack in a bunch of these calcium ions and pack them together so that you can get a muscle contraction to happen. And in your nerves, it's going to be packing in a bunch of sodiums and potassiums and packing them into a specific area to get that nerve impulse to fire. So those are the reasons or the times where you would need that active transport to force those ions to be in close proximity to each other. Now that takes us to hyper, hypo, and isotonic and the rest of our packet. And that is actually going to be our discussion when we come back face to face for our next class on the 28th. So please make sure that this packet comes with you because we are going to finish up the second half of the packet next time we come together. Please do not forget that you have your quiz over the anatomical terms. It is just the anatomical terms. You had the front sheet, the back sheet. It was in the area of 42, 43 terms between the front and the back, but that is the only thing that is going to be on the quiz. It is not any of our other lectures or any of our other material that we have covered, just those anatomical terms. If any questions come up between now and the 28th, please send me an email and I will get those answered for you. Otherwise, have a great evening or a great day wherever you're at, and I will see you on the 28th.